Hey, good morning. Um, Before we get into our lecture this morning, I had left a little post-it note for myself on the computer that says no quiz. So I decided, because this is another week where it's a short week and Friday is a holiday, um, a big portion of the class has their recitation sections on Friday. And so I decided, even though originally I wanted to have a quiz and recitation, the more I thought about it, the more I thought it'd be complicated on how to handle the quiz to make it fair for the Thursday students as well as the Friday students. So I figured, you know what, the easiest thing to do is just to not have a quiz this week. And then the material that the quiz would have been on, this is talking about things like biological catalysts, enzymes, that kind of stuff that we've been discussing the last couple of lectures. We'll just save that for next week's quiz, and that'll be sort of in there as part of some of the other material that we're learning now as well. Okay, and so I decided that after I had met with the TIs earlier this week, and so I informed them that I changed my mind and we're not going to have a quiz. All right, so don't worry about that. Okay. Let's continue talking about energy usage in the cell. We had been talking about glucose as the primary fuel source for living cells, and we said that glucose has to be converted into a chemical form of energy that is then usable by the cell, right? And that is ATP. So let's back up and start discussing how it is that glucose can be broken down and converted into ATP in order to be able to distribute the energy in those chemical bonds throughout the cell to all the different proteins and biochemical pathways that are expecting ATP as the format, right, or the form of the energy. So we'll talk about this process of glycolysis today. We'll also talk about fermentation and gluconeogenesis. So this cartoon, helps get us in sort of the right frame of mind, I think. When we talk about glucose, here's a molecule of glucose, C6H12O6. We've been using that as an example of catabolic processes where you can break apart bonds in a molecule like glucose to be able to tap into the energy that's represented by all of those electrons that are participating in these bonds, right? All of those electrons, even though this is a stable molecule, those electrons, if given the opportunity, would really prefer to be engaged with other atoms in different arrangements that put them at lower energy orbitals. Right? We said that fortunately, the molecules like glucose that we depend upon for life are metastable. That means this glucose molecule will stay together indefinitely, right? I don't just mean a few hours. I mean a molecule of glucose could stay together for 10,000 years or more, right? Because those electrons at least are stable in their bonds. And that's good, because if glucose just fell apart by itself all the time, you couldn't rely on it, right? It's kind of like building if you have a set of playing cards, right, poker cards, and you build like a little house with them. We've done that before. It's kind of fun, right? I mean, it's metastable, but not really. In other words, sure, you can carefully build your little house of cards, right? But the moment a draft comes in from the window or somebody bumps the table, it comes crashing down, right? So a house of cards is not very metastable, right? It's just metastable enough to hold together. So fortunately, molecules like glucose, right, are more metastable than that, right? But importantly, they can still be broken down, right? Because the opposite would also not work, right? If you actually, for example, glued all of your cards together, with crazy glue or something, or Elmer's glue, whatever, right? And then, yeah, okay, great, you have this house of cards, but you're never going to be able to take that apart again, right? So the opposite would not work either. So that's why life evolved to make use of molecules that have that just right metastable quality, that they are stable enough to be moved around and transported, and they won't just fall apart by themselves, but at the same time, when needed, their bonds can be broken, just as long as you have appropriate biological catalysts like enzymes that fit the right shape, right? And can do something to actually help encourage these bonds to break. So in a molecule of glucose, look at this equation here, this chemical formula, which we've looked at already. So a molecule of glucose plus 6 molecules of O2, right? If they're encouraged to react together and all of the electrons that are involved in all of these bonds have the chance to kind of break existing bonds and sort of find or re-engage in bonds that are better, meaning lower energy, so the electrons are happier, right? If this is allowed to go to its conclusion, you end up with 6 carbon dioxides and 6 waters, right? And we said that the amount of energy released by this then, by all the bonds on the left rearranging to all the bonds on the right, that difference in energy from potential energy at the start all the way down to whatever kinetic energy is kind of released, you get 686 kcals per mole of glucose released by that. And we said in biological reactions, this is as far as we take it, right? Because you could say, well, yeah, but how come I can't break this down further, right? You can if you sort of go overboard and put way more energy into this, right? For example, if you were to heat this up with a flame, right, and get all those electrons busting apart, right, just as long as you have oxygen available on the left, right, those oxygens also bust apart and all the electrons come together, you would go to carbon dioxide and water. But if you keep heating this up, you know, then you might even break this down. Eventually, you break the carbons and the oxygens apart, right? And you'd get just elemental carbon left over, right? The black carbon, right? But in our cells, we don't take it that far, right? And importantly, remember that the combustion of glucose depends upon the availability of oxygen in order to burn, right? You know, I think ever since we're little kids, I say this a lot in this class, ever since we're little kids, because, you know, through my many years of doing science, I've often thought about, you know, the way we think about the world when we're children, you know, our view of things, and how, you know, as we learn more about science, there are fundamental changes in the way that we view things. So one of the things that I always thought when you're a little kid, you know, you say, oh, you need to have oxygen because oxygen burns, right? Something won't burn without the oxygen, as though it's the oxygen itself that somehow blows up and releases heat, right? I mean, that's the impression that you get. But the reality is, no, the oxygen doesn't like blow apart releasing heat, right? The oxygen is there because the oxygen is a very greedy electron acceptor. Right? It happily participates in encouraging the electrons, for example, in the glucose bonds, right? Giving them someplace else to go. The oxygens will happily take them, and therefore, right, you get the release of energy every time you go from a higher bond to a lower bond. And if everything is allowed to happen, then you get this reaction. And you get a lot of heat released. And if this is just happening without anything to harness the heat, that's your flame. Right? That's why there's so much energy released in the combustion of glucose. But without the oxygen, you're not going to get the glucose burning with a flame, right? Will the glucose still fall apart? Yeah. If you heat up glucose with extreme temperatures without any available oxygen, right, the glucose will fall apart because, of course, all the electrons are still freaking out and eventually they bust apart. You're just not going to get the release of heat like a flame. You go through a process that's not called combustion. You go through pyrolysis, right? Where chemically the reactions happen and things break down, and you end up with things like methane or elemental carbon, things like that at the end. A lot of it is just released as water, right? So if you heat it up a lot, in the absence of oxygen, you do start releasing some water from it, so you dehydrate. That's why a pile of sugar, right? A pile of sugar, if you try to light it on fire, it won't burn, right? Because in a pile of sugar, there's not much oxygen like in that pile with all the granules stacked up together, right? Whereas something like a fluffy marshmallow, which is basically still that glucose or that sugar, right? But now it's puffed up, so there's lots of air in it, right? That will burn and release flame because there's lots of opportunity for oxygen to kind of mingle in there, right? All right, so before I go off on a tangent further, you know, you're like, oh, okay, we get it, Dr. Shen, right? Okay. So the point is what, right? When you look at this equation, you all know from basic chemistry that a balanced chemical equation like this has all the atoms on the right present already as all the atoms on the left. But when you show it visually, like with these colored spheres here, I think this is a nice way to appreciate that, you know, for real, right? All of the red balls on the right here representing oxygen, all of the black ones representing carbon, all of the white ones representing hydrogen, they are all present on the left here. So this is really the ideal rearrangement of all of these, going from higher energy bond states to lower energy bond states. Okay. Again, inside of our cells, we're not talking about applying heat like a flame in order to do this, right? We have catalytic enzymes that step by step break down the carbon-carbon bonds systematically, each step of the way liberating a little bit of energy, right? And importantly, each step of the way being able to harness that energy so it is not lost all as heat, but instead it can go to doing something else. For example, going to form a molecule of ATP that then can float away and represent that energy that's now being transported. Just like we said, if you have coal, fossil fuels delivered to an electrical power plant, right, the power plant can tap into that energy in the coal and convert it into electricity that can then be distributed to your neighborhood, to your room, and to the electrical outlet on your wall where you plug your phone in, right? Because your phone is expecting electricity as a way to recharge. It is not expecting a lump of coal. Right? So that's great. Hopefully you understand, yeah, so that's why oxygen is important, right? If you really want to get the most energy out of glucose, you got to have oxygen to participate in taking these electrons. But hey, not so fast, right? Because when life first started off on Earth billions of years ago, there was very little atmospheric oxygen, very little O2 floating around, all right, for life to actually learn to use to make these processes. So Earth is about like 4.6 billion years old. And for the first billion years, right, there was no atmospheric oxygen. Our atmosphere was filled with all kinds of nasty stuff like methane, carbon dioxide, you know, different types of sulfuric compounds, things like that, right? But very little atmospheric oxygen. Therefore, when life first began, you know, and Mother Nature first began to experiment with how life was going to work, right, it had to come up with ways of making energy molecules like glucose in the absence of O2. Right? That's why life evolved first to be photosynthetic, right? Because you do have energy raining down on Earth in the form of the sun, which we still enjoy to this day, right? Every day, the sunlight represents energy that's beaming down to Earth that can be tapped into. You can harness that energy and use that to actually make molecules that, in a way, store that energy in the form of those chemical bonds that the energy was used to make, right? So early life had plentiful carbon dioxide, and, you know, our planet was blessed with having liquid water, right? So, you know, we had oceans of water that were forming. So life evolved to use carbon dioxide, water, taking the energy from the sun to kind of break those bonds apart and rearranging them into glucose, right? Remember, the last couple of lectures, we've looked at these cartoons where we compare the catabolism of glucose from C6H12O2 plus oxygen down to carbon dioxide and water, right, which is what our cells do. But then on the other side of the cartoon, it was what plants do, right? Plants take the CO2 and the water, take the energy from the sun and build it back up into glucose. And in the process, right, you end up liberating a molecule of O2 because that's the waste product of photosynthesis, right? So glucose metabolism then, in order for these primitive bacteria to use that glucose that they were making, and now they want to be able to break it down and make things like ATP from it, they had to do it anaerobically, meaning without the help of atmospheric oxygen. Right? So this is a nice artist rendition of what primitive Earth looked like, just at the dawn of life first evolving. So this is maybe like, 3 billion years ago or so, where we had liquid water oceans, right? The sky was filled with all kinds of gases like CO2, sulfurous compounds released by the volcanic activity of our Earth still sort of cooling down. And in these oceans, you had the formation of photosynthetic bacteria, right? So these large sort of stump-looking things are these towers of primitive microbes that are photosynthesizing, right? They're taking carbon dioxide, they're taking water, and they're making sugars, right? And what are these bubbles that are being released from them? Oh, that is the waste product of the photosynthesis. That is atmospheric oxygen, right? So already you begin to see that although life had to start off in oxygen-free conditions, because the waste product of them doing photosynthesis is oxygen, you begin to start having the buildup of more and more oxygen in the atmosphere. All right, so this process of primitive microbes learning how to take a molecule of glucose and being able to at least tap into some of the energy in that glucose to make some ATP anaerobically, right, is this process of glycolysis. So glycolysis is ATP generation without the help of O2. Right? So we're going to focus today looking at this. So glycolysis happens in 3 phases inside of cells. Here, this very simplified cartoon, I use this simple kind of way just to sort of keep the focus here, right? Here's glucose, right? So it's 6 carbons in a ring. We typically think of this way, right? So phase 1 of glycolysis is sort of manipulating this glucose to basically make it sort of a symmetrical molecule, right? And the way that cells do this is they actually have to use up 2 molecules of ATP to get the phosphates off of them, right? So 2 molecules of ATP get used by the cell here in an enzymatic reaction, right? And that helps to kind of make this symmetrical shaped glucose here. Right? And then at the end of phase 1, this symmetrical glucose, it's no longer called glucose technically, right? Because it has these phosphate groups on it and it's been rearranged. But you can already see how this can be split into 2 halves, right? So then phase 2 is taking each one of these half molecules, each one of these 3 carbon half molecules, and oxidizing it. So you will have enzymes that then oxidize this half molecule here. And in the process, you're able to take that ATP, take that phosphate group off and make a molecule of ATP, all right? In the process, the enzymes that are involved in doing this, right, they're taking electrons off of this molecule, right? That's the oxidation that's happening here. Those electrons need to go somewhere. We learned last time that many enzymes that move electrons around don't end up just being stuck with the electrons themselves, because that wouldn't be very efficient, because in order to kind of reset that enzyme, you'd have to then have another enzyme that comes and takes those electrons off, and so on and so forth. So we learned last time that life evolved to use small little sort of expendable little molecules as electron acceptors, right? So the enzyme will bind to something, it will transfer the electrons from one molecule to this electron acceptor. And we learned last time that a very important coenzyme, right, is NAD plus. So from here on in, I often refer to in my cartoons as the NAD as being sort of like this basket, right, that can then be loaded up with the electrons, and it becomes no longer NAD plus, it becomes NADH, right? And then this electron carrying basket can then just float away, and the enzyme that's doing this oxidation of the glucose can still work freely, all right? So you get 1 molecule of ATP from that. Phase 3 then is you take what's left of this 3 carbon molecule and you kind of manipulate it around again through some more oxidation steps to be able to generate another molecule of ATP, right? And then what we end up with at the end is this 3 carbon sort of leftover bit, which is pyruvate. So remember, each one of these phase 2 and phase 3 steps here is being done to each one of the half molecules that come from phase 1. So that's why I put 2 times and this whole thing in a bracket here, right? So therefore, you end up getting 1, 2, 3, 4 molecules of ATP out of this part of the cartoon. But remember, you spent 2 molecules of ATP just to allow this happen at the beginning, right? So therefore, the net overall gain of glycolysis is you get 2 ATPs out of a molecule of glucose. Right? Let's look at these phases in a little bit more depth. All right. So let's get away from this very simple cartoon and let's go ahead and actually see what's going on. So let's start with phase 1 with the glucose coming in. And you're like, oh my God, you know, yeah, that you liked that simpler cartoon better, right? Yeah, I know. But let's learn about this, okay? So here you have the glucose molecule. It's being represented here not as a ring, but here it is linearized. Okay, so the 6 carbons are present there. All right, so you start off with glucose. So there will be a series of enzymes that take turns at different steps along the way, working on their particular substrate or their particular intermediate molecule in this pathway. So first off, you need to spend a molecule of ATP, right, in order to start to get a phosphate group on there. Right, so you go from glucose to glucose 6-phosphate. All right, so that's not very tricky because it's glucose, but with a phosphate group stuck on the 6th position. Right, then another enzyme, an isomerase. Remember, an isomerase, the ACE part, you know, it's an enzyme, right? And isomerase means it's an enzyme that actually is able to switch different molecules between their isomeric forms. Remember, isomer means what? The same chemical formula, just the atoms arranged differently, right? So glucose is converted by a glucoisomerase enzyme into fructose 6-phosphate, right? So fructose is the same chemical formula as glucose. It's just a different arrangement, right? Then another enzyme over here, right, a fructokinase, comes and is able to, with the help of spending another molecule of ATP, right? So this ATP gets converted into ADP by this fructokinase enzyme, right? And in the process, that kinase, oh, right, we learned this, right? It's called a kinase. So we said, what does kinase mean? An enzyme that transfers phosphate groups onto a target, right? So in this case, it takes that second phosphate off of this ATP and plops it here, and you end up with fructose 1,6-biphosphate. Again, you know, if you just looked at these names, you'd be like, oh man, I have to know this. But, you know, you think about it and it's not that hard, right? It's a fructose with 2 phosphates, so it's a bisphosphate at positions 1 and 6, right? Then at the end of phase 1, you have this enzyme over here that essentially now cuts this molecule in half. So this fructose 1,6-biphosphate, this is on my simpler cartoon, the kind of more symmetrical looking molecule intermediate, because you can imagine cutting this in the middle here and having 3 carbons on this side, 3 carbons on this side, right? So the bottom part is glyceraldehyde 3-phosphate. The upper part is this dihydroxyacetone phosphate, but you can have an enzyme that easily converts that into glyceraldehyde 3-phosphate as well, right? So the end of phase 1 is you end up with 2 of these glyceraldehyde 3-phosphates, or G3Ps. Right? Let's start with the G3P on our next slide, where we look at phase 2. Right? So here's phase 2. Here's the glyceraldehyde 3-phosphate. Right? And keep in mind now, right, we say this is happening twice because we're just looking, this is the same thing is happening to both of the half molecules, both of the G3Ps that are coming from phase 1. So the glyceraldehyde 3-phosphate, it gets oxidized. So you get the addition of another phosphate group to it, right? And in the process, you have this enzyme over here, this glyceraldehyde 3-phosphate dehydrogenase, that removes a couple of electrons off of it, right? And that's the process of oxidation. And it uses one of those electron-carrying baskets, right? So an NAD basket would have floated by here and be bound up by the enzyme at the same time that the enzyme is binding to the glyceraldehyde 3-phosphate, and it transfers electrons into the basket, and the NADH basket then floats off, right? So that leaves you with this molecule here, this 1,3-bisphosphoglycerate, right? And then one of these phosphate groups then can be handled by this enzyme here, right, which is another kinase that goes ahead and transfers, right, that phosphate group onto an ADP, making an ATP molecule, right, leaving a 3-phosphoglycerate at the end. All right. Then phase 3 starts with this 3-phosphoglycerate. So here it is over here. Right? And through a series of enzymes, it basically just gets rearranged. All right, you can see really what's happening is the phosphate group is just getting kind of pushed around onto different parts of the molecule. Why? So that eventually when you get to this structure over here, this structure can be worked on as the substrate by this enzyme over here, this pyruvate kinase, right? Which then can take this phosphate off of here, add it to an ADP that comes floating by, turning that ADP into an ATP, right? So this is your second ATP that's made if you were following just one of the glyceraldehyde 3-phosphates that came off of the original glucose molecule. But because the glucose molecule got split into half, right, all of this stuff in phase 2 and phase 3 is 2 times, right? So in reality, this is how you make your 4 ATPs, right, out of this process, but you have to subtract out the 2 ATPs that went into this process to start off with, right? So that's why the net gain is 2 ATPs from this. And the leftover molecule is this pyruvate, right? And you look at this and you're like, well, I don't get it, right? Because this still looks like half of the glucose. I mean, it's still 3 carbons. You know, and it's kind of like, well, can't you do something more with this? Well, the point is that early life evolved to stop here. There's not much more that early life figured out how to be able to break this down further to get more energy out of it, right? Because in order to do so, you would need more oxygen molecules to be able to work on this, right, as ways of getting electrons pulled off of it. So the end of glycolysis is the formation of this pyruvate. That doesn't necessarily mean that early microbes did nothing with the pyruvate, right? It's not like they just let the pyruvate go, because one important thing to think about is in order for that glycolysis process to happen, you needed to have those electron-carrying coenzymes participating, those baskets like NAD+, that can then be a source of where enzymes can dump electrons, right? And you're filling up these baskets. So if you fill up these baskets, then you can't use them anymore. Right? So early life evolved a way to kind of add on some additional steps after glycolysis, after the formation of pyruvate as the waste product of glycolysis, to at least use pyruvate as a mechanism to help empty those baskets so that those baskets can then be used again. And that is the process of fermentation. Hey, right, we all know fermentation, right? Because fermentation is important in things like beer, making certain types of cheeses, yogurts, in many types of foods, traditional foods. Fermentation is a part of this, certainly in a lot of Asian foods, right? You think about kimchi or even types of like fermented tofu and stuff like that, right? But what is fermentation, right? Why does fermentation happen? Right? Fermentation evolved as a way for primitive anaerobic microbes to get some additional use out of the pyruvate, not to make more ATP from it, right? Because they didn't have the ability to do so, but you could use pyruvate as a way of emptying those electron baskets that you filled up. So let's look at this here. Here's pyruvate from glycolysis, right? So early microbes evolved this enzyme, which is lactate dehydrogenase. And what it does is here comes, one of those filled electron baskets, an NADH, right? This enzyme, the LDH, the lactate dehydrogenase, can take the electrons off the basket. So the basket then becomes emptied again as NAD+. And that NAD+ can go back and help out in the glycolysis steps, right? Because it's now free to accept more electrons. So what you're basically doing is you are reducing the pyruvate down to lactate, right? So this is lactate, right? So it's a reduced form of pyruvate, right? Similarly, another pathway is starting from the pyruvate, right? And going through a couple of enzymatic steps to make acetaldehyde, which releases some carbon dioxide from it, and then using alcohol dehydrogenase, right, to empty the electron basket, right, that was filled by the glycolysis in the first place. You're now able to get those electrons out of the basket so that the basket is NAD plus again, and it can go back and help out with glycolysis some more. And you've then reduced the acetaldehyde down to ethanol, right? So that's why lactate and ethanol are really the end product of anaerobic mechanisms of glycolysis. And these represent simply additional steps to be able to at least use the pyruvate to kind of free up those coenzyme electron carrying baskets so that they can go back and help out. Right? So let's talk about lactate, for example. Here's glucose. Through the process of glycolysis, you're able to get 2 ATPs out of the glucose through those 3 steps of glycolysis that we started off the first half of this lecture talking about. And importantly, you ended up filling one of these NAD plus electron accepting baskets, right? Turning it into an NADH. Right? And you ended up with pyruvate. Right? So you're not going to be able to get more ATPs out of this pyruvate without the help of oxygen. But what you can do is you can use lactate dehydrogenase to take that filled electron carrying coenzyme, empty it out, right? Get those electrons back onto the pyruvate, turning it into lactate, right? And you've freed up the NAD plus to go back and help out with more glycolysis, right? Pretty clever, right? So that lactate, of course, is the reason why fermentation is useful for different types of foods. That's what gives cheese the tanginess, right? Or yogurt, that tanginess. So lactate then is really the end product of pyruvate being worked on or being reduced in order to help support more glycolysis happening upstream. So many anaerobic microbes, right, they will start with glucose or lactose in their environment, right, and they will go ahead and do this glycolysis and fermentation because that's what they're designed to do. And so we get cheeses and yogurts that way. Importantly, the muscles in our body, right, the muscles in our body that are catabolizing glucose to get ATP out of them to power our muscles. Right? If we end up working our muscles too hard and we don't have enough oxygen getting to them to keep additional ways of getting more ATPs out of the glucose, right, then our muscles end up building up a lot of lactate in them. Right? Because importantly, even though I didn't expressly say this, right, I'll say it now. Even though this glycolysis pathway is what early life on Earth came up with in anaerobic conditions, right? You know, the way evolution works is once it starts on the path of coming up with certain systems to do things, right? Suddenly, if things change and you have, oh, now we have a lot of oxygen, we don't need to do this glycolysis anymore. It doesn't work that way, right? You build upon pre-existing systems. So to this day, our cells, when they're first handling glucose, right, and beginning to tap into it, our cells still do glycolysis first in order to be able to get a couple of ATPs out of it, right? It's just we also have additional ways of getting more energy out of the glucose rather than just being stuck with the pyruvate as the end product, right? But under extreme conditions, when our tissues are not getting enough oxygen and we don't really have the ability to get more energy out of the glucose, we end up building up a lot of lactate, right? So you know this. So a lot of people say, hey, that's why your muscles get really sore because there's a buildup of lactic acid, right? I actually don't know if that's really true because, to be honest, I don't know if really enough lactic acid builds up in your muscles to really cause like pain, right, or inflammation. But, you know, whatever, that's what people say. And at least it gets them thinking about the fact that, yeah, you're having a buildup of lactate if you can't really efficiently get enough oxygen to your muscles, right? So that's why lactate fermentation is really an important part of how our tissues function, right? To this day, even though, yes, you thought we were just talking about glycolysis because we're talking about primitive microbes and you're like, who cares, right? You should care because the point is our cells to this day still start off by doing the glycolysis steps, just like we've learned, right? Because that's what evolution came up with, to start off with. So yeah, in our muscles, we can have a buildup of lactate, but we have evolved ways of using that lactate. So our lactate can go into our blood and can end up going to the liver, where we have different enzymes in the liver that can kind of piece together little scrap pieces of lactate to try to make some additional glucose molecules out of it, right? To be able to at least recycle parts of the spent glucose to try to make some more glucose molecules. And we'll learn about that at the end of this lecture. What about alcohol fermentation? So we said microbes that do glycolysis, right, and end up with pyruvate as their waste product. You could do the lactate fermentation, right, to try to empty those electron baskets, ending up with lactate as your waste product. Or, you know, some microbes preferentially do ethanol fermentation, right? So from the pyruvate, they have an enzyme here that begins to break down, or not break down, but modify the pyruvate, altering it into acetaldehyde, right? And in the process, it releases carbon dioxide. So that's why this carbon dioxide is what you experience when you have things like yeasts, for example, that are blooming, I guess they call it, when they're making dough, right? And you let it rise. It's because you're having the production of carbon dioxide being formed by these microbes as part of them doing something with the pyruvate. Right? And then we have this alcohol dehydrogenase, the ADH, that can then actually take the electrons from the electron basket, the NADH, right, and reduce the acetaldehyde, and it turns it into ethanol, right? And so that's your waste product at the end, right? And in the process, you freed up the NAD plus to go back and help out in glycolysis some more, right? So the ethanol really is like the waste product of the microbes, right? It really is like the poop of the microbes, right? And so, yeah, great. It's kind of ironic that we've learned to enjoy drinking the ethanol so much. Right? One thing I wanted to point out was, remember, at the end of last lecture, when I was first teaching you to think about how complex biochemical pathways involving intermediate molecules have enzymes along the way at different steps in order to facilitate, right, efficient use of these pathways to do things like detoxify things. So we had looked at ethanol as the starting point last time. And we say when you're drinking the ethanol, the poop of the microbes, right, your body needs to get rid of it because, A, it's toxic, right? So we said the enzyme that converts ethanol to acetaldehyde is this alcohol dehydrogenase. Right? But you're like, wait a minute. That was going in the opposite direction, though. How come in this case it's going in this direction? Remember, enzymes, certain enzymes function by just getting different reactants together to be able to accelerate something happening. And that something happening could be, we usually think of it as it binds some sort of reactant and it breaks it apart into two products. But right, if there's no energy really required in doing this, then the process can go in reverse. You can have separate products that are come together and actually formed by the enzyme, right, into something else. So the arrows go both ways. Right? So that's why, in terms of what the microbes care about, they tend to use the ADH to convert acetaldehyde, reduce it down to ethanol, right? Because they're trying to free up these electron carrying baskets. In our body, if we're on purpose having this ethanol in us where it doesn't belong, right, we need to break this down. So the ADH in our bodies favors working in the other direction, taking the ethanol and converting it to acetaldehyde. And then if you look back to the last lecture, you would have a different series of enzymes from this point on, right? So this would take a detour and maybe go down here somewhere where we break the acetaldehyde down to, other smaller metabolites, eventually to CO2 and water. Right? Okay, so if glycolysis is all about taking glucose and putting it through this process to get a couple of ATPs out of it, glucose, remember, is one of several different possible types of sugar structures that something like a microbe or something like your cells would have access to. Remember that something like table sugar, sucrose, is a disaccharide of glucose and fructose, right? or maltose, like in malted beverages, or lactose, right, in milk, is a disaccharide of galactose and glucose. So when these get broken down by different enzymes that break the disaccharide into separate monomers, right, glucose is the one we typically think of as being the important one, right? That's why classically the glycolysis pathway starts off by assuming you already have your glucose, right? And that feeds into phase 1 of glycolysis, right? This whole thing is just phase 1 of the 3 stages of glycolysis. But consider this, right? Mother Nature doesn't want to be wasteful. If you're breaking down these disaccharides, then you have the other sugar monomer that was part of it. For example, the galactose that comes from the lactose can be worked on and converted by different enzymes into glucose 1-phosphate. And glucose 1-phosphate then, so this is now glucose with a phosphate group on the first carbon, right? It's just a matter of moving that phosphate group from the first carbon to the 6th carbon, and you end up turning it into glucose 6-phosphate, which is already part of glycolysis, right? Likewise, fructose that comes from the breaking down of table sugar, right? The glucose has to be fed through over here, but the fructose, right, can be fed in at this stage because you have fructose and you have some enzyme that works on it and it turns it into fructose 6-phosphate, right, which is already part of this phase 1 of glycolysis anyhow. So different sugars and different components can enter glycolysis through different sort of entry points, right? Look at this, glycogen and starch. Remember, glycogen and starch are simply long strings, polymeric chains of glucose, right? So obviously, glycogen and starch can feed into this glycolysis pathway as well. And so, so that involves enzymes that will break off one of the glucoses from, let's say, glycogen, right? And when that's broken off, the form of that glucose that's broken off is the glucose 1-phosphate, right? Which then has to be converted into glucose 6-phosphate, and that's where it enters the pathway, right? Now you might say, wait a minute, upstream of this pathway was the first time that ATP had to be invested in this, right? You needed to spend a molecule of ATP to convert it to convert glucose into glucose 6-phosphate. But if you already have glucose 6-phosphate coming in from over here, and this didn't require ATP, wait, are you sort of somehow now cheating, right? Because does that mean that if you have glucose that enters from this side as glucose 6-phosphate, that you actually only end up spending 1 ATP to do glycolysis? And therefore the net gain is actually not 2 ATPs, but 3 ATPs? No, right? Sadly, you never get something for nothing, because what you don't see in this picture, but hopefully you can think about, is ATP was used in the process of actually having glycogen to start with, right? So the end result is you still only get a net gain of 2 ATPs. Look here, glycerol, right? The 3-carbon glycerol that we've talked about in terms of being like the staple that holds together things like phospholipids or triglycerides, right? That already has 3 carbons in it, right? So that could already feed into the glycolysis pathway, but it would enter over here, right? Because you don't have to go through all this fancy converting of glucose into 3 carbons. You already have a 3-carbon molecule to start with, right? So that's why things like the glycerol from things like different types of lipids and things like that can also feed into the glycolysis pathway. All right. So to summarize then, glycolysis and fermentation together is how primitive life came up with a way to be able to use the glucose that was being made through photosynthesis of assembling carbon dioxide and water together into this high energy glucose molecule, right? In the absence of available oxygen, right? It couldn't do that wonderful thing, that cartoon at the beginning where we have fresh air and all the atoms can disassemble and reassemble, right? If there's oxygen available, right? Early life didn't have that. So it came up with glycolysis, right? And glycolysis involves taking a glucose molecule, right? Doing various manipulations of it through 3 different phases, and you end up getting 2 ATPs out of it and a pyruvate left over, or actually I should say 2 pyruvates, right? From each 6-carbon glucose, you end up with 2 3-carbon pyruvates as the end result of glycolysis. And fermentation evolved as sort of an add-on to this, not to get more ATPs out of it, but to at least use the pyruvate as a way to kind of take those electrons back from those baskets, so that the baskets, these NADHs that were loaded up during glycolysis, could be emptied and then recycled as NAD pluses that would go back and help out with the glycolysis again. All right. So I mentioned that of the fermentation pathways, ethanol really is like the most waste sort of, you end up with ethanol, and ethanol is just, even the microbes don't want it, right? So they are just getting rid of the ethanol. We are the dumb animals that come along and drink it, right? Okay. But so that's really just waste, right? So the lactate, on the other hand, can be used to build other cellular molecules. And I mentioned our liver has the ability to take all these scrap lactates that our muscles are generating when they don't have enough oxygen, and put these scrap lactates back together in a process called gluconeogenesis to make a few more glucose molecules. So what's that about, right? So gluconeogenesis takes that principle of having a chain of enzymes, that are going in one direction. And if those enzymes don't really require energy to go and make the pathway go in a particular direction, from reactant to product, then that chain can be reversible, right? So gluconeogenesis in many ways is the reverse of glycolysis. Let's look at this figure here, starting with glucose, right? And this is just the phase 1 of glycolysis that takes you all the way down, through glycolysis, and you end up with glyceraldehyde 3-phosphate, right? That's the molecule that's at the end of phase 1 of glycolysis, right? Many of these enzymes can work in both directions, right? Some of these enzymes cannot. For example, the enzymes like this glycolysis enzyme 1 and glycolysis enzyme 3, they needed to break ATP to ADP in order to make the reaction happen, right? So those are not necessarily reversible. Right? So you would have other enzymes that kind of take their place at those steps, right? But the other steps that don't require energy being invested, these enzymes can work in both directions. So what we're talking about is skeletal muscle. We're taking glucose. We're working out really hard, maybe harder than the oxygen demands of our muscles can keep up with, right? So you end up building up more and more lactate from just having this be the product of lots of glycolysis happening. That lactate goes into our blood, it goes into our liver, and our liver has enzymes that can then basically convert that lactate, piece things back together into pyruvate, and then the pyruvate back to glucose, right? Importantly, you might say, wait a minute, right? Does this mean like we don't even have to eat more glucose because we can constantly just recycle lactate back into glucose? How does that work? No, right? It doesn't work that way, right? The lactate represents sort of the scrap pieces of leftover glucose, right? And so anytime you're dealing with assembling or cobbling together scrap pieces to make something whole again, right, you're usually not going to have a very efficient time doing it. The example I give is, hey, you guys know, my wife is a wonderful baker, right? She bakes things like cookies, all kinds of things, scones, and she also makes biscuits. So, sometimes she rolls out a sheet of dough to make biscuits, and she rolls it very thin, like in a rectangle rolling pin. And then she has like the little mold or the little, I don't even know what you call this little tin kind of biscuit shaped cutting thing. I don't know. Right. And she can stamp out maybe like 12 biscuits from that nice flattened out rectangle. Right. And then she takes the scraps, the little margins that are left over. She will go ahead and work that together and flatten that out. But maybe she'll get maybe a couple more biscuits from that. Right. So that's the point, is that glucose, as your body's breaking it down and you have all these scrap pieces of leftover lactate, right? It's not like you get one glucose molecule back from every one lactate, right? It's a diminishing returns. So why do we do it? We do it because there are certain conditions where tissues in our bodies might be really starved for glucose, right? Think about things like your brain, for example, right? Your brain is probably like the top priority in your body to keep happy and healthy and functioning, right? Because if your brain doesn't work, right, it doesn't matter that your foot doesn't work or the muscles in your biceps don't work anymore, right? So under stressful conditions where there's low glucose, gluconeogenesis can be used to break down and take scrap other molecules and try to assemble some glucose molecules that can then be delivered to places like your brain that are being starved of glucose, right? So it's kind of like a last resort sort of thing, not something that we can actually really depend upon, right? All right, let's go ahead and stop there today.